The second part of the chemical senses is to talk about olfaction. So we'll start with that. And we can see that we're going to start in this bottom-up kind of way, talking about how the stimulus is based on chemicals, that we are dealing with odorants that are odor-inducing substances. They are small, volatile, hydrophobic molecules. And there's all my pieces and words that they're small molecules that are repellent to water. They're volatile, they're gaseous, they're in the air, and they come up and they hit our olfactory receptors. Not all chemicals with these properties produce any smell, and a really good example is carbon monoxide, but there are quite a few things that they are chemicals that are in the air, gaseous, repellent to water, but they do not produce smell as well. Functions of the olfactory system. One is to be this warning system. I mentioned with taste that these are gatekeepers. With taste, we are actually taking those chemicals into our bodies to get some kind of substance out of them, right? We're eating this, we're drinking this, we're putting it in our bodies. The olfactory system, it allows us to smell things before having to put them in our bodies. So spoiled food, leaking gas, smoke, we can smell these warnings from a bit farther away. We also have a response to pheromones. So the definition for pheromones is any chemical produced by a species that influences the behavior of others in the species. A study that's been not replicated by other people and who have complained about the statistics of Stern and McClintock. But then their original study was published in the 70s, and they went and did a replication in 1998 and found the same and had the same findings, where I feel like the way they handle their statistics, the way people complain about it doesn't make a lot of sense. But so, because what they were looking at was the timing of the release of the luteinizing hormone. So if I'm going to get this in the right direction, and let me do this experiment description first. So they had donor women and recipient women. And the donor women, they went and they took underarm secretions from the donor women when they were, we'll start with this, when they were at their ovulatory phase of their menstrual cycle. And then they went and they put that under the nose. They treated it so it wouldn't smell like anything, and then they put that under the nose of the recipient women and told them to leave it there for at least 6 hours. And then what they found was that this sped up the release of luteinizing hormone and shortened the recipient woman's menstrual cycle. They also went at another time during the follicular phase of the menstrual cycle, took the underarm secretions from the donor women, wiped them under recipient women's noses, and told them to leave it there for 6 hours. And what they found was that this lengthened the time before that surge of luteinizing hormone, and it lengthened the recipient woman's menstrual cycle. So this whole idea that we have this menstrual synchrony, it does appear that there's a reason for that, and that it's pheromones that are influencing us. There are other studies looking at pheromones now because it is controversial. We see this. We see in other animals that they're responding to pheromones. They also have this vomeronasal organ that has these receptors in it. It looks like our humans' vomeronasal organ is vestigial and does not have any receptors in it. And so the suggestion is that these pheromone receptors are kind of littered throughout and within, like the olfactory receptor region. But so that's, I guess, part of the reason why it's very controversial. But there's other studies that have looked at this, and I'll just use a couple of examples. One where, so I'm just going to use one example. One where they took underarm secretions from women, or no, I'm sorry, they took sweaty t-shirts from women, and had men smell them. And if the woman was, it was during her ovulation, the men released more testosterone than if it was not during the woman's ovulation. They also, in a separate experiment, rated the smell of the sweat as more pleasant if she was in her ovulation or ovulatory phase than when she was not. So it looks like we do have these reactions to other people's pheromones. And finally, the olfactory system provides all of this differently for different animals, and they usually distinguish between macrosmatic animals and microsmatic animals, where we are microsmatic. Humans, we have a less keen sense of smell. We have less of our cortex devoted to smell. It's less crucial to our survival. This is no longer really how we need to find food, right? This is no longer how this isn't how we find mates necessarily. That might be questionable to some extent. Macrosmatic animals, they are using that sense of smell to find their food, to find their mates, to smell their predators or their prey. So dangers as well as food. Talk about the detection of odors. We're looking at detection threshold, one of these thresholds, and they decided to go with a forced choice where they would have a cotton ball originally with an odorant on it, some amount of an odorant, and another cotton ball with no odorant on it, and they would ask which has the stronger smell. And if the person was about 75% or higher, they would say the person can smell that odorant at that level. And if they were under 75%, because it's a 50-50 chance kind of thing, they would say that the person was unable to smell that particular odorant at that particular level or that threshold. They did this because if you go back and think about how we're measuring these thresholds, and we talked about signal detection theory, well, apparently for smell, we have a particularly unique and individualistic kind of motivation, I guess is the word, for when we feel like we can smell a smell. Some people, they have a teeny tiny maybe something's out there and they're like, yes, I smell it. And other people, they have to really smell this and waft it in, and then they're finally like, oh, okay, I think I smell that. So apparently that's why they went to the forced choice to get to that detection threshold. They have found that rats are more sensitive, cats are more sensitive than humans, dogs are more sensitive than humans. And if we look at what's going on, oops, I did this in a funny way. If we look at what's going on with those different groups, humans are estimated to have somewhere between 10 million to 20 million olfactory receptor neurons, so those receptors themselves. And you can see that those other animals have more receptors than we do. So we can only get down to where we are, where one molecule of an odorant influences one receptor to fire, but some of the animals have more receptors. And one of my favorite little fun facts, another fun fact about elephants, is that they can smell water about 19.2 kilometers away, which I can't even smell the water in my book bag. So I can't even smell water when it's right next to me. I'm not really sure how they do that, but clearly they need that ability since they're often living in arid deserts and places that there's not much water. I'm providing some more thresholds, specific thresholds, which I did at the very beginning, and I said you don't need to know these. I'm just showing really here that different odorants have different thresholds, and I think that makes sense. So we're looking at parts per billion, and that T-butyl mercaptan, that one is what they add to natural gas to give it a smell, and so we want to smell that, right, at very few parts per billion. The other one I like to just jump to is acetone. 15,000 parts per billion is what we need in order to smell it. Acetone is what's in nail polish remover that gives it its smell. And that's one of those things I can walk into a room and someone's taking off their nail polish and I feel like I'm breathing it down my throat. So I was a little surprised at how much higher how much higher that is. So one of the people who have looked into this is William Kane, these thresholds, and he was looking at actually difference thresholds. Can you tell there's more of an odorant on this, or that they are different from each other? And originally they were using cotton balls, and he found a difference threshold at about 19%. But he realized that there were differences in how the cotton ball is being held. There are differences in airflow. It is what we call kind of messy situation, or that it has a lot of situation noise. And so using the olfactometer, which he strongly started suggesting, right, that we start using when we are making measurements of the olfactory system. And if you look at what the olfactometer is, they are precisely controlling so that we know exactly how much humidity and exactly how much of the odorant a person is receiving up their nose. And they got this to, this difference threshold dropped to 11%. If we look at recognition thresholds, it is different. We need a concentration that is increased by as much as a factor of 3 above the detection threshold for people to be able to recognize odorants. And they still don't do the greatest job. So apparently we can detect, or the estimate is that we can detect as many as 1 trillion different odors. As you can imagine, this is an estimate because we can only measure so many odorants at a time on certain people. And what they would do is they would make these slight changes, and then they estimated, well, it must be about 1 trillion. This is another one of those things that it's kind of funny what's controversial, but this also is a little bit controversial, and when you're in the field. But then recognizing them is much more difficult, and it looks like part of the issue is that it is our memory for the odor name where we are often lacking, that we're not able to get to the odor name. And this I'm going to talk about a few times as something that's really interesting, because we never do this kind of like, what is that? What is that? And someone says, oh, that's a dog. And you're like, oh, it's a dog. But we do that with odors. And he's talking about this, I think it was called aquavit, this particular alcohol. And they were drinking it, and they were making these guesses. Oh, I think it's anise. I think it's orange. I'm not sure. And then someone reads the back and says, oh, it's caraway. And then everybody has this just transformation. They're like, oh, it's caraway. And it gets this label, and that actually influences the perception of the odorant or the smell. And I know I'm talking about flavor here in a lot of ways, but so we will talk about how, for our experience of flavor, odor has a huge influence. We're not just dealing with those basic taste qualities. And if I used flavor at all when I was talking about taste, that was my mistake. But so yeah. And a major puzzle of, the puzzle of olfactory quality is that we can have a similar molecular structure like the ones up in A, where there's very little difference, right? Here's this oxygen versus these 2 hydrogens. Otherwise, this is a similar molecular structure. And the odorants are very different. So we're comparing a musk to something having no odor. We can have down in B these really different molecular structures, which those two have so much different, I can't even go through it very easily. And they have a similar odor. They smell similar. They both smell like pineapple. So I ask, how is this different than in vision? I usually actually have, how is this different in vision and in hearing? Right? We've talked about a number of sensory modalities, and they're really different than this. So you can pause and think. And this is to me a neat and interesting puzzle. So typically when we think about vision and we look at the electromagnetic spectrum, those 600 to 700 or so, we have these different reds, and then we move down to orange, and we move down to yellow. So we have these things that are similar along the electromagnetic spectrum, and they appear similar in the world. If we move down all the way down to the short wavelength lights, we get down to blue and indigo and violet. Same with hearing. When we're up at the high pitches, those pitches sound similar to us. They share aspects of they're similar to each other as far as the stimulus and the physics, and we perceive them as similar. So olfaction is very different. The latest edition of Sensation and Perception, they have added this section, this short little bit of information about anosmia. It was being written during the pandemic, and so this was a thing, right, that people really started looking into because of how many people lost their sense of smell as a result of COVID. But so they talk a little tiny bit about congenital anosmia, so people who are born with a lack. So A is not having, and anosmia would be being able to smell. People who are born not being able to smell. It's interesting some of what they handle and deal with. So our sense of smell does keep us safe from certain things. There's a lot of social aspects to our sense of smell. And when we start talking about how smell goes right into those memory systems, it seems pretty complicated, and it would be interesting to know more about how this plays out with people who are born with this inability. Anyway, so they also talk about anosmia as a result of COVID. So when people were losing their sense of taste and smell, it was really mostly because of their sense of smell, right? Again, flavor is both taste and smell, but smell is a huge part of flavor. And what it looks like was happening is that SARS virus, it attached itself to these angiotensin converting enzyme 2 to those proteins, and that those exist in the support structures in the olfactory epithelium. So some of this should sound a little familiar. We talked about the pigment epithelium for vision, that was really providing enzymes and nutritional support for those receptors. That's kind of what's happening with the olfactory epithelium. So those ACE2s, those proteins were not in the olfactory receptors themselves, but they were in those support cells. And finally, they talk really briefly and give us some a nice figure that shows how anosmia is an early symptom of Alzheimer's. And since it is stimulating the hippocampus in some of those memory areas, it could be a symptom that shows us somebody is more likely to have Alzheimer's, although it's an early symptom of other things as well, like Parkinson's and Huntington's. But it could be that lack of smell, that inability to smell, could be somewhat causal, as it is an early symptom, and again, that would be stimulation to the hippocampus. So it's not completely clear, but it can be an indicator, which is helpful to know. So if we get to the neural code for olfactory quality, we have the olfactory mucosa. This is an area that is kind of made up of the olfactory epithelium, those underlying tissue and blood vessels, glands, bundles of axons from the olfactory neurons. You can think of it as the retina of the nose. It's moist up there to really drive. So those small, volatile, hydrophobic molecules basically get driven to the cilia on the olfactory receptor neurons. You can think of it as protecting the olfactory epithelium, but it's really the olfactory epithelium is in there, but it's being protected, and it allows those odorants to dissolve. The olfactory receptor neurons, they are like rods and cones in a number of ways that are interesting. So they contain molecules that are sensitive to odorants, like rods and cones contain visual pigment molecules that are sensitive to the electromagnetic spectrum. They are sensitive to a range of specific stimuli, which is what we saw, right? With those long wavelength cones, they had the stimuli that they responded the best to, but they were sensitive to the stimuli around that as well, right? They responded some to some of the ones close, some of the nanometers of wavelengths that are close by. So we have a similar kind of sensitive to a range of specific stimuli. And they are made up of proteins that cross the membrane of the receptor 7 times. So we're back at that interesting kind of, that protein molecule is crossing 7 times. Don't know why this is a thing, but this is a thing. And what's interesting, one more piece of this that's interesting, they're different in the way that we have rods and we have 3 types of cones. So we basically have 4 types of photoreceptors, but for olfactory receptor neurons, it looks like there are more like something between 350 and 400 different types of olfactory receptor neurons. We have something like 300 pieces of genes, so genes that are coding for olfaction, which is another reason we see lots of differences between people, because we can have certain olfactory receptor neurons that respond to certain ranges of stimuli. We can have different ones. We can have different numbers of different olfactory receptor neurons. And then I have this other species comparison that there are estimated to be about 1,000 different types in mice, which suggests that while we're smelling or able to differentiate about 1 trillion different odors, they're at least twice that much. Okay, so as I'm talking about olfactory receptor neurons, I will do my transduction. So this is the magic. This is the magic that happens.